Thank you very much for the introduction. My name is Alencar, and today I will present you this research that was performed in Biogenomic Laboratory in the Federal University of Santa Maria, Brazil. And this research was performed using human adipose-derived stem cells obtained from lipoaspirates that are highly cytogenotoxic, susceptible to hydrogen peroxide. Well, as we know, the fundamental unit of our body are our cells. And the development of all our tissues and all our body is associated with embryonic stem cells. And these embryonic stem cells will start this process of body formation and tissue development using these two ways: proliferation process and differentiation process. However, our adult cells are not immortal because we have a process that's called apoptosis. That's a physiological process and is normal, and we need this process because we need to keep the balance of our cells and the number of our cells. But at the same time that we need the apoptosis process, we also need a cellular renovation, and this cell renovation process will be done for our stem cells, adult stem cells, using the same process of proliferation and differentiation. So then, we can say that our adult stem cells are able to repair our tissues when necessary and to keep the balance of our body, to keep the number of our cells, and then to keep our homeostasis. On the other hand, we also know that in the last decades we observed a significant increase in the number of people around the world with chronic degenerative disease. And this specific point increased the number of researchers around the world searching for new alternatives to treat this chronic disease. And this search was much more potentialized when we start to work with adult stem cells. So then, was only possible to start to do it when we improve a lot of our cell culture conditions and our molecular biology methods. Our first studies with stem cells start with embryonic stem cells, but sometimes it's more difficult to work with this type of cells. So then, with detection of specific receptors for adult stem cells, we start to work with this type of cells, and then the first results using this type of cells show us that it's much more easy to isolate adult stem cells, and it's much more easy to keep these cells alive in cell culture conditions. And the first studies also show us that adult stem cells are able to do a positive response for different disease, for example, metabolic disease, heart disease, and cancer. So, when we have our stem cells inside to our body or in vivo, these stem cells don't have senescence process because our adult stem cells are able to keep the cell renovation, and then only when these stem cells receive a signal to start the differentiation process is that this adult stem cell will change the morphology and will form a new differentiated cell. So then, this differentiated cell is start the process of aging, and then after a long time, a specific time, will be senescent. But when we are working with adult stem cells in cell culture conditions, we have sometimes this process that's called the premature senescence. And this premature senescence is able to decrease the proliferation and the differentiation process capacity. And why this premature senescence will happen? Sometimes when we are keeping the cells in cell culture conditions. The cell culture condition is an artificial condition, and then this point can increase the cellular stress and then start this premature senescence. So, what's our problem? Our problem is that sometimes when we are working with stem cells for a specific point, for example, a transplant, or when we need the stem cells to make an experimental model, we need a high number of cells. So, with this, we need to keep the stem cells for a long time in cell culture conditions, and this long time will increase the possibility of contaminations, and then will increase the possibility of senescence. So then, sometimes when we have the number of stem cells that we need to work, a lot of these cells are senescent. And it's interesting that stem cells, as in vivo as in vitro, when receive the signal to start the proliferation and differentiation process, these cells have an increase on the intracellular levels of hydrogen peroxide, because this hydrogen peroxide works as a signal to start this both process. So a lot of research around the world start to search if we can use the hydrogen peroxide as a molecule to increase the proliferation capacity of our stem cells and then decrease the long time in cell culture conditions. But currently we don't have on the literature description if it's safe to use this molecule as a new alternative to it. So then the objective of our study is exactly this, is to test the effect of hydrogen peroxide at different concentrations on acute cytogenotoxicity and oxidative stress using adult stem cells obtained from human lipoaspirates. So then, to do it, we first obtained 3 different samples of lipoaspirates, and after enzymatic digestions, we used only the stromal phase, and we keep this stromal phase in cell culture conditions. Using the stem cells at 40 passage, we plated these cells, and then after with concentrations from 1 to 1000 micromolar of hydrogen peroxide, and after 2 hours of exposition, we start to perform our experimental evaluations to contemplate our objectives. So then, the first assay that we performed was to evaluate the cytotoxicity effects of these kinds of treatment. And to this, we used this reagent that is specific to quantify the double-strand DNA. And with this, it's possible to measure the levels of mortality. And to confirm the results for this first assay, we also performed the caspase 1, 3, and 8 determination using a ELISA system. We also performed here this assay, the dichlorofluorescein diacetate. That's an assay where it's possible to measure the reactive oxygen species. And to available the lipid peroxidation, we used T-bars, and to measure the activity of superoxidase dismutase and catalase activity, we use different kinds of colorimetric assays. And here, our main assay was this one, DNA common alkaline assay, that was used to measure different levels of DNA damage. And we also performed here, to this study, the cell morphology evaluation to evaluate if our treatment are able or is able to change the morphology of our stem cells. So then, in this first picture here, we have in this first one, the first day of stem cell isolation, and here our cells in the passage number 4. And using these cells, with our treatments with different concentrations of hydrogen peroxide, we observed that our treatments with this molecule is able to increase the levels, increase significantly the levels of mortality of adult stem cells from the concentration of 1 micromolar, and especially on the concentrations of 300 and 1000 micromolar, we observed 100% of mortality. So then, the next steps for this study, we performed only using the concentrations until 200 micromolar. And here, in this picture, we have our stem cells of our negative control without treatment, and this one is our stem cells with the concentration of 200 micromolar. And then we can observe that our treatment is able to change the morphology of our stem cells. So here, these graphs, we can see that the treatment with different concentrations of hydrogen peroxide is able to increase the levels of caspase 1, 3, and 8, and then confirm that these treatments are able to increase the mortality levels of our cells. And here in this table, you observed a significant increase on the levels of ROS, increase in the levels of lipid peroxidation and catalase activity that was dose dependent. And here we also observed an increase in the levels of the activity of SODI, superoxide dismutase, but was not dose dependent. And in this graph, we observed that from the concentration of 10 micromolar of hydrogen peroxide, we had an increase on the levels of the DNA damage. So, the literature described us that the hydrogen peroxide can activate our adult stem cells and can increase the capacity of proliferation and differentiation. But currently, this research don't describe to us that it's safe to use this molecule. So then, our results show us that when we increase the levels of reactive oxygen species, we can do a lot of damage in our adult stem cells, because we observed 100% of our cells going to die from the concentration of 200 micromolar. And this was confirmed using the measuring the levels of caspase 1, 3, and 8. And as expected, we also observed an increase on the levels of ROS. We observed a significant increase on the levels of catalase. That probably is because the function of this enzyme is to neutralize the hydrogen peroxide. We observed increase on the levels of activity of superoxide dismutase, but not dose dependent. And here, especially with this result, we can say that we observed a significant increase on the levels of DNA damage and morphologic alterations. So then, we can say that maybe hydrogen peroxide can activate our stem cells and can increase the capacity of proliferation and differentiation. But in the same time, this kind of treatment is able to increase the levels of cytotoxic effects and genotoxic effects. So then, I would like to thank to Biogenomic Laboratory, and especially to my supervisor, Ivana da Cruz. And I also would like to thank to Pharmacology and Toxicology Laboratory from the University of Toronto, especially to Dr. Ana Andreasa and Dr. Gustavo Scola. Thank you very much for your attention.